Hi, my name is Sohail and today I am going to talk about morphology of chromosomes. In this lecture, I will focus on P arm and Q arm of chromosome, primary and secondary constrictions, heterochromatin and euchromatin, DNA, gene, allele, locus and classification of chromosomes. Chromosomes are rod-shaped filamentous bodies present in the nucleus which become visible during cell division. They are carriers of genes. Chromosomes are not visible in active nucleus due to their high water contents but are clearly seen during cell division. Chromosomes are major carriers of genetic information in eukaryotes, and chromosomes they are present inside the nucleus. Chromosome means colored body and chromosomes are virtually colorless. The term refers to their ability to be stained by certain dyes. Chromosomes are composed of DNA and protein, and this protein is known as histone. In chromosome, 40% DNA and 60% protein is present. What is the difference between chromosomes and chromatin? As we know, the chromosome, it is composed of DNA and protein that is called histone. So the DNA is packed with the help of this protein that is called histone to form a thread-like structure which is called chromatin. So chromatin, it further condenses to form chromosome. This means that chromatin is lower order of DNA organization, whereas the chromosomes are higher order of DNA organization. A significant amount of RNA is also associated with chromosomes because chromosomes are the sites of RNA synthesis. So this is messenger RNA which is synthesized from DNA, and the process in which messenger RNA is synthesized from DNA, that is called transcription, which takes place inside the nucleus, and DNA is present in chromosome. So that's why mRNA and other types of RNA are also associated with chromosomes. Each chromosome contains a single DNA molecule which runs uninterrupted through the entire length of chromosome. A typical chromosome contains 140 million nucleotides in its DNA. So DNA, it is made up of nucleotides, and nucleotides are the monomers because they are basic repeating units, and there are 4 types of nucleotides, that is adenine, guanine, cytosine and thymine, and 140 million nucleotides are present in the DNA of a single chromosome. If we think that each nucleotide as a word and the amount of information an average chromosome contains would fill about 280 books, and each book contains 1000 pages and each page with 500 words. So there is tremendous amount of information present on a single chromosome in the DNA. In a cell, DNA is compacted and it is fit into a much smaller space. Chromosomes, they can vary by length, the number of base pairs in DNA molecule, position of centromere, and genes occur at a specific locus or location. That is, it is always found at the same position on the same chromosome. What is locus? A locus is the specific location of gene on the chromosome. A locus is a specific location of DNA sequence on a chromosome, like a genetic street address, and the plural is loci. The place in chromosome where gene is located, that is called locus. So locus is the home of a gene. As we know, the chromosome is composed of DNA and protein, and the length of DNA which is present in a single chromosome, it would be 5 centimeter. If we could lay out the strand of DNA from a single chromosome in a straight line, it would be 5 centimeter. Fitting such a strand into a cell nucleus is like cramming a string the length of football field into a baseball. Each mitotic chromosome consists of two sister chromatids that are held together at a waist-like constriction that is called centromere, and the portions of chromosomes that are not in the centromere itself are called chromosome arms. This is the constriction which is called centromere, and this length it is called chromosome arms. And these two chromosomal arms, they are called sister chromatids because they are attached to the same centromere. Now the chromosome is divided into two parts with a constriction in the middle, and this constriction is known as centromere. So a centromere is a constricted region of a chromosome that separates it into short arm which is called P arm and the long arm which is called Q arm. Now what is P arm and Q arm? P arm of a chromosome is a short arm, and P comes from French petite, meaning small. So all human chromosomes, they have two arms, that is P arm and Q arm. And these two arms, that is P arm and Q arm, are separated by this constriction, that is primary constriction known as centromere. It is the point at which chromosomes are attached to the spindle fiber during cell division, and the part of centromere which is attached to the spindle fiber, that is called kinetochore. And the symbol Q was chosen for the long arm simply because it followed P in the alphabet. When chromosomes are represented as a karyotype or ideogram, where each chromosome is arranged in such a way that the P arm is positioned above the centromere and Q arm is represented below the centromere, and centromere is the primary constriction. So this is the karyotype of human genome, and in each chromosome P arm is directed upward and Q arm is directed downward. What is secondary constriction? Secondary constrictions mark the locations at which the nucleoli are assembled. As we know, nucleolus is present inside the nucleus. Nucleolus is singular, nucleoli is plural. And secondary constrictions that mark the locations at which nucleoli are assembled. So it is associated with nucleolus during the interphase of cell division, and take part in reorganization of nucleolus at the end of the cell division. Some parts of secondary constrictions are also known as nucleolar organizing region. The important genetic information is retained because the chromosomes, they have protective end caps. So these are the end caps which are present at the end of the chromosome, and end caps are called telomeres that do not contain protein coding genes, and telomeres that consist of short non-coding guanine rich DNA sequence that repeat many times. The terminal ends of chromosomes are called telomeres, which prevent the two chromosomes to attach with each other from their ends. Now the difference between heterochromatin and euchromatin. As we know, chromatin is composed of DNA and histone protein. So nuclei, they contain two broad classes of chromatin, that is known as heterochromatin and euchromatin. In non-dividing cells, chromatin fibers are loosely packed in some regions and densely packed in other regions. The loosely packed regions are called euchromatin, and the densely packed regions are called heterochromatin. Hetero stand for different. Heterochromatin is highly condensed throughout the cell cycle. Cell cycle that consists of interphase with three substages, that is G1, S and G2, and after the interphase there is mitosis and cytokinesis. So throughout the cell cycle, heterochromatin that is inactive in transcription. And what is transcription? It is the synthesis of messenger RNA from DNA. It's mean the genetic material which is present here in the DNA of heterochromatin that is not expressed. that is not transcribed. So mRNA is not synthesized in the region of heterochromatin. Euchromatin, which is less condensed part where the genes are active, and RNA is synthesized from DNA. So transcription process that takes place in case of euchromatin. So mRNA is synthesized in euchromatin region only. Generally speaking, chromosomes are not visible under light microscope. So during cell division, chromatin fibers, they coil, undergoes a process which is called condensation, forming a thick rod-like structure which is called chromosomes, and they are visible under light microscope only in dividing cells. But in non-dividing cells, that's present in the form of chromatin material. Chromosome is made up of DNA and protein which is called histone, and DNA contains genes. So what is a gene? Gene is a fragment of DNA which is responsible to control a particular character of the body. So genes are heritable factors that control specific characteristics, and genes are part of DNA, genes are fragments of DNA. And what is allele? Alleles are different forms of a specific gene. And what is locus? Locus is the place on chromosome where gene is present. So it is a home of a gene, or it is a street address of a gene. Now homologous chromosomes are the chromosomes that have same structure, and in somatic cells which are called body cells, homologous chromosomes are present and the cells are said to be diploid, that is 2n. In homologous chromosome pair, one of the chromosome is paternal, that is inherited from the father, and the other is maternal, that is inherited from the mother. Size, homologous chromosomes are the same size and structure, and they carry the same genes at the same loci, but alleles carried at each locus may be different. Based on the position of centromere and length of chromosomal arms, chromosomes are classified into 4 groups: telocentric chromosome, acrocentric chromosome, submetacentric chromosomes, and metacentric chromosomes. Telocentric chromosome that have a centromere at one end of the chromosome, and humans, they do not possess telocentric chromosome, but they are present in certain other species like mice. Acrocentric chromosome, which is offset from the center, leading to one very long section and the other very short section, that is called acrocentric chromosome. And human chromosomes number 13, 15, 21 and 22 are acrocentric. Some metacentric chromosomes, they have unequal length of 2 sections, and human chromosome number 4 through 12 are submetacentric. Metacentric chromosome, they have centromere primary constriction in the center, and the 2 sections are of equal length, and human chromosome number 1 and 3 are metacentric. To sum up, P arm and Q arm of chromosome. P arm of chromosome is the short arm of chromosome, and P, it comes from French word petite, meaning small. So all human chromosomes have 2 arms, the P arm which is a short arm and the Q arm is a long arm. P arm and Q arm are separated by primary constriction that is known as centromere, and the part of centromere which is attached to spindle fibers, that is called kinetochore. Secondary constrictions mark the locations in nucleus where nucleoli are assembled. So it is associated with nucleolus during interphase of cell division and take part in reorganization of nucleolus at the end of the cell division. Heterochromatin and euchromatin. Heterochromatin is the part of chromatin which is highly condensed and the genes are not expressed, and euchromatin is the active part of chromatin where transcription process takes place. As we know, the chromosome is composed of DNA and protein, that is called histone, and DNA, it contains genes. Gene is a fragment of DNA, gene is a section of DNA, and gene is heritable factor that control a specific characteristic of the body. Allele is the alternative form of a gene, and locus is the place on chromosome where gene is present, that is called locus. Locus is singular, loci is plural. Based on the position of centromere and length of chromosomal arm, chromosomes are divided into 4 types, that is telocentric chromosome, acrocentric chromosome, submetacentric chromosomes and metacentric chromosomes.